Hi everyone. So this is the second problem solving session for the course Basics of Biology, and we're going to continue from where we left last week. So let me just present my screen. Yeah. So this in this session we're going to speak about cell biology. So we'll first start with what is cell. A cell is defined as the smallest basic unit of life that is responsible for all of life's processes. So what are the characteristics of cells? Cells are the structural and functional and biological units of all living beings. A cell can replicate itself independently, hence they are also known as the building blocks of life. Each cell contains a fluid called cytoplasm. Cytoplasm is enclosed by a membrane and contains several biomolecules like proteins, nucleic acids, and lipids. And so before we move on to what are cells and what are the characteristics of cells, we'll first see how were the cells discovered. So the process of discovery of cells. So the cells were first discovered by Robert Hooke in the year 1665. So he observed a piece of bottle cork under the compound microscope and he saw minuscule structures that reminded him of small rooms. So cell basically refers to the rooms that he had seen. And next, we have Anton von Leeuwenhoek, who observed cells and their motility under another compound microscope with higher magnification. So, Leeuwenhoek was able to look at the motility of cells, and because he could look at them being mobile, he said that these are alive entities and he called them animalcules. And then in the 1883, we had Robert Brown, who provided the first insight into the structure of the cell when he looked into the orchid cells and he could find the nucleus and he described it, because of which the structural component of cells was able to be understood better. So based on this, now let's talk about characteristics of cells. So cells provide structure and support to the body of an organism. The cell interior is organized into different individual organelles surrounded by separate membrane. And every cell, as I have already mentioned, has one nucleus and membrane-bound organelles in the cytoplasm. And so cells can be of different type based on what kind of organism we are looking into it. So first classification is prokaryotic versus eukaryotic cells. So the prokaryotic cells are smaller in size, roughly around zero point 0.2 to 2 micrometer in diameter. And for the eukaryotic cells, they are larger. So they are around 10 to 100 micrometer in diameter. And the most important feature differentiating these two cells are the presence of nucleus. So in prokaryotic cells, you will not have a proper nucleus. You will rather have a nucleoid-like structure. There won't be any membrane to it. While for the eukaryotic cells, you can have true nucleus, which will have nucleoli and nuclear membrane. And other than this, in the prokaryotic cells, you will not have membrane-bound organelles. In the eukaryote cells, you will have membrane-bound organelles, for example, lysosomes, Golgi complex, endoplasmic reticulum, and so on. And then the next feature is about flagella. And eukaryotic cells will have complex flagella, while the prokaryotic will have simpler. And similarly, you will have about difference in the cell wall. So in the prokaryotes, you will have a cell wall, which is mainly composed of peptidoglycan. And in eukaryotic, if it is present, it will be very simpler in the structural component. And then other than that, you have the compositional differences in the ribosomes. So ribosomes are where you have protein synthesis. So the ribosomes have building blocks and the components of ribosomes differ. For example, in prokaryotic, the smaller size ribosome will be of 70S, S being sweet work unit. Basically, it's the centrifugation unit. And in the eukaryote, the larger size will be of 80S and the smaller size will be of 70S. And as we have discussed previously, cell division in prokaryotic is binary fission, while in eukaryotic, they are based on mitosis. So let's describe prokaryotic cells in a bit more detail. So prokaryotic cells are also the single cell microorganism, and which include the bacteria, archaea, and photosynthetic cyanobacteria. They consist of single membrane, and that's why the reactions occur in the cytoplasm. And as we have seen in the previous slide, they lack organelles like mitochondria, endoplasmic reticulum, and Golgi bodies. So if we have to go over the slide, the structure in detail, so I'll just start the pointer. Just give me 2 minutes. Yeah. So here in the prokaryotic cell structure, you can see that there is a capsule, which is the outer protective covering for the bacterial cells in addition to the cell wall. Cell wall is just beside it. So above the layer of cell wall, you have the capsule. And this capsule is also helpful in the moisture retention, protection to the cells, and also in attachment of the cell to nutrient surfaces. Then beneath capsule, you have cell wall, which is again the outermost layer of the cell, giving the shape to the cell. So the cell wall supports the structure of the cell. Then you have the cytoplasm, which will compose of mainly all the enzymes and organelles and salts. Then you have the plasma membrane or the cell membrane. This layer surrounds the cytoplasm, and here on the plasma membrane there will be proteins that will regulate the entry and exit of substances into the cells and out the cells. And then you have pili, these small hair-like structures, which help them to attach to any surface for the bacteria. And then you have flagella, which helps in the movement of these cells. Then you have ribosomes, if you see here in the blue colors. So these ribosomes, as I have mentioned, they are involved in protein synthesis. And then you have plasmids. So plasmids are extra chromosomal DNA here, and we'll come to this plasmid in a bit more detail. And then you have the nucleoid region. As I have mentioned, they don't have nucleus, so they have this nucleoid region in the cytoplasm as a spreaded material. Now, we talked about plasma membrane, and on the basis of plasma membrane, you have two categories of cells in the bacteria. They are all called gram positive and gram negative, and it is very important because based on this structure, you have a very popular or common experiment which is called gram staining, which can help you to identify classes of the bacteria. So the sample that you have, whether it is gram positive or gram negative, can be understood by this experiment, and the basis of this experiment is based on the structure of the plasma membrane. So if we just go over here, on the left of your screen it is gram positive and it is gram negative. So how to understand that? First, if you just look at the cell wall, the plasma membrane is here and the cell wall layer is here. So in the gram positive bacteria, you see that the cell wall is quite thickened because of the peptidoglycan layer. So the peptidoglycan layer is quite thick here. And how does it become so thick? So it is formed of NAG, which is N-acetylglucosamine, and NAM, which is N-acetylmuramic glucosamine. And these polymers are actually linked by beta-1 fold linkage. So this cross-linking makes it very stable. So this structure becomes very stable. And sugar polymers here are attached to the peptide chain composed of amino acids like alanine, glutamic acid, lysine, and alanine. And this peptide chain, as I have mentioned, they are cross-linked to each other, which makes this cell wall very strong. But in gram-negative bacteria, if you compare, you see this layer very, very thin. And then on top of this layer, you have the lipopolysaccharide layer, which contains the porins. Porins are the proteins which are responsible for movement across the cell. So with this idea in your mind that in gram-positive cells, you will have thick cell wall, and in the gram-negative, you will have a thinner cell wall. So this is the protocol of the, or these are the steps of the experiment, which is called the gram staining. So we'll start with this. So this is how you see the cells on the slide when you are doing the experiment. This is the final step, but this is the ongoing process. So let's say you are given a smear, a sample, and you make a smear onto your slide. You fix it. So you fix by heating the cell. So once you fix it, it looks something like this. The next step is putting crystal violet on the slide. With crystal violet, what will happen is the peptidoglycan layer and the crystal violet will, like this, they will stain the cell wall. So the cell now look purple to you in color. Then you add iodine. Now what iodine does is iodine forms a cross-linking with the crystal violet and it forms a complex. Now imagine if it is a gram-positive bacteria which will have a stronger peptidoglycan layer, or a broader peptidoglycan layer, it will have more cross-linking and it is stable in structure. But for the gram-negative bacteria, where the crystal violet complex is formed, but it is not able to adhere to the cell wall because the peptidoglycan is very, very small or very thin. Now what do? You wash the cell with ethanol. Now these complexes, which are very stable and gram-positive, retain the complexes and still look purple. But in the gram-negative bacteria, complex is washed off. The purple color now goes away and it becomes white in color. And then what you put is a counter stain, which is safranin, which is red in color. Now the cells which were already purple color will have no longer space to hold the red color, so the color of safranin. But in the gram-negative bacteria, the red color will be attached to the cell wall now, and when you now look in under the microscope, you will see gram-positive bacteria as purple and gram-negative bacteria as red. So that's the idea of identification of these microbes under this slide. Now I had mentioned about plasmids. So what are plasmids? Plasmids, you will be, it has a very unique property of being extra chromosomal and circular DNA, but it can also be in linear form. But why is this so important and how it is a focus of research is because it is used in molecular cloning. Generally, you will have plasmid, which is used as a vector. Now, in vector, you put your gene of interest and you express it in another cell. So, let's say you want to express a certain gene in a certain plant. Now, first you need to have that gene expressed. That protein needs to be taken out and put into your desired organism. But first you need to produce that protein outside the cell that you want to insert. So you want to have a vector where you can attach this protein's gene, and then you want to put it into the host so that it can amplify there. Now, for that to amplify and to also introduce itself into the host, plasmids are used because they are very amenable to this. Now, why? We'll see this. So first of all, plasmids are extra chromosomal circular DNA molecules, and these plasmids can exist in different species and they can be same in structure, and they are transferred from one bacteria to the other bacteria via horizontal gene transfer. And the unique property of these plasmids are they have their own origin of replication, so ORI. So when you will read about DNA replication, you will realize that the origin of replication is very important to initiate DNA replication. Now, because plasmids have their own origin of replication, they can replicate themselves and form their own copy. Now, you have to go back to the idea that you have taken your gene of interest and you have inserted them. How? I'll come to it. But you have inserted that into the plasmid, and now, because it has the origin of replication, it will replicate and replicate, making copies of itself. And in the process, the desired gene of interest that you want to express in larger number will also be amplified in a larger numbers and be expressed. So that happens there. Now, why and how it is important for the bacteria? Other than the fact that it will have its own origin of replication, it will have antibiotic resistant genes. It will have many other genes which will be useful for survival of the cells. So it will have genes like ampicillin resistance. And this ampicillin resistance is what is used as a screening method when you are doing your cloning studies. And other than that, you have several sites which can be cleaved by restriction enzyme. So for example, here, you can see that there is an ampicillin resistance here and a tetracycline resistance here, and you want to put your gene of interest here. And so what you do is you cleave this with EcoR1 and BAMH1. So this part, because it can be cleaved by these enzymes, they are cleaved off, and you put your gene of interest here. Now what does that do? Because your tetracycline resistant gene is disrupted, this plasmid is no longer resistant to tetracycline. So when you grow this cell in a medium which consists of ampicillin, it will grow because it will have an intact ampicillin resistant gene. But when you grow it in a tetracycline medium, it will not grow because it has lost its resistant gene. That's why you can screen if your gene of interest has got inserted into the vector. And that's how you do your screening for cloning. And in the bacteria, you can also have different kinds of or different forms of plasmid. So they can be circular, which is the native form. They can be supercoiled. The DNA gets super wound to each other, and because of which the size gets shorter. So they are here. And then you have linear, which becomes so here the DNA becomes supercoiled. Here the DNA becomes linear, and as a result, from supercoiled DNA to linear DNA, the linear DNA is bigger in size, so it is above in the gel. And nicked is just one opening, so it becomes linear, but it is just it's still held here at some point. So because of which this has biggest size. So these are the different forms of plasmid that you can see on the gel. Now, these are all about prokaryotic cells. Let's move on to eukaryotic cells. For eukaryotic cells, uh, you have them as more complex. So the eukaryotic cells are more complex and they contain many membrane-bound organelles to perform specific functions. So they contain nucleus. As I mentioned, the prokaryotic cells will not have nucleus. They will have nucleoids. But in eukaryotic cells, you have nucleus, which are isolated, which can be isolated from the cytosols and enclosed in a wall-defined plasma membrane. So they will have membrane-bound organelles. Now, in eukaryotic cells, also there are two differentiation. They can be animal cell and they can be plant cell. They also differ across several characteristics. For example, shape. Plant will have a defined shell because they will have a rigid cell wall. Animal cell will not have a fixed cell shape. And as I mentioned, that it is because of the presence of cell wall. So plants have cell wall because of which their shape is rigid, while the animal cells do not have this cell wall. Now, vacuoles. Plant cells will have large vacuoles, while in the animal cells, there won't be any vacuole. Chloroplast, as you know, chloroplast is for photosynthesis, so it will be there in the plant cell but not in the animal cell. For food storage, plants store food as starch, while animals store them as glycogen. And centrioles. Centrioles will be present in animal cells. We will see what is the use of it, but in plant cells, they won't be present. So, we'll first move on to cytosol. So cytosol and cytoplasm are more or less same, but there are finer differences between them. So as you can see in the images also, this outer green portion, the lighter green is cytoplasm, while the darker portion is cytosol. And so what is the difference between these? Cytosol is a fluid present in the cell membrane, while cytoplasm is a cell component present inside the cell membrane. So here it is a component. So the cell cytoplasm will have many organelles attached to it. Cytosol is just a fluid. Cytosol is composed of soluble ions like water and water soluble molecules, but cytoplasm will be rich in enzymes, water, lipids, nucleic acid, all the important macromolecules. In cytosol, reactions take place in prokaryote because prokaryotes are very basic in their cells in structure. But in cytoplasm, you will have larger cellular activities like cell division and glycolysis. And then in cytosol, you have transportation of molecules and signal transduction. In cytoplasm, you will have important functions like cytokinesis, nuclear division, and signal transduction. And as you can just imagine from the fact that cytoplasm will have many enzymes and proteins, so the diversity is high in cytoplasm and less in cytosol. So with these factors in mind, let's just go over these points once again. So the cytoplasm includes the cytosol and all the other organelles. Cytoplasm, however, does not include the nucleus because nucleus has its own membrane, so it is separated from the cytoplasm. Cytosol is not uniform because it is just a fluid structure which takes shape according to the cell, shape of the cell. And protein complexes and cytoskeleton gives it the structure. And as we have already gone over the functionality of both cytoplasm and cytosol, they have their distinct functionality each. Now, after cytoplasm, we'll move on to nucleus. So, as I have consistently mentioned, the nucleus have their own membranes, which are referred to as nuclear envelope. And the why is nucleus so important? Because in nucleus you have the chromosomes. Chromosomes have the DNA, and DNA is the genetic unit of any cell. So they provide the genetic information required for creation of different cell components. And also, nucleus is the site of DNA replication and site of transcription. But translation is outside in conjunction with other organelles. So we'll first go to nuclear envelope. So if you just go back here, you will see that these are the nuclear envelopes. So we'll study this structure in detail to understand how the export and import of the proteins or any molecules takes place across the nuclear membrane. So the nuclear pores are tiny orifices that facilitate communication between the nucleus and the cytoplasm. So if you just look into this is the nucleus and the outer membrane is the cytoplasm. So the movement of the proteins or movement of the molecules are actually between the cytoplasm and the nucleus. And the component or the cellular component or fluidic component of the nucleus is called the nucleoplasm. So the nucleocytoplasmic exchange is carried on by at the tiny orifices or the nuclear envelope. And here, small ions and molecules that are less than 5,000 Dalton, they move freely across it. Well, if there are proteins which are greater in size, then they have active transport. What do you mean by active transport? You mean that there is a facilitator and there is an exchange of ions that drives the energy, because of which you can get those molecules inside or outside the cell. And the nuclear pores have a diameter and thickness of 120 nanometer and 50 nanometer. So if we just go in the details of the pores, you can see that there are these bridges. So these bridges are there in the nucleoplasm layer also, and in the cytoplasm layer also. They are held to each other in this basket like formation. You have this cytoplasmic ring on the outside and this ring which is on the inside, and then you have these structural component that give support to this basic filaments, and across this pore is where the exchanges take place. Now we'll move on to mitochondria, which is a very important component of the cells. And as you would have heard it multiple times, that mitochondria is the powerhouse of the cell or power unit of the cell. Why? Because we have gone over it last time also, that it is responsible for ATP synthesis. So it is why it generates power for the cell. But other than that, also mitochondria has many other features. For example, it's a double membrane bound organelle. So here if you see the structure, you will see that it has a double membrane. So this outer layer, this is called the outer membrane. Then you have this thinner green, which is called the inner membrane. And then you have these the intermembrane space where you will see the finer components. So, and this is where the biochemical reactions take place. So, how does this mitochondria term came about? Mitochondria composes of mitose and chondrions. So, mitose is thread and chondrions are granule-like. So, we'll come to the granules in a bit. You'll see what do you mean by granule. And it was described by Richard Altman. So, here, if you see, these are the intermembrane space, the lighter green portion, and then you have this crystal-like structure. These are matrices, and on all of these boundary, you have ATP synthesis. So the ATP synthesis is present all across this membrane of this matrix, and you have ribosomes and mitochondrial DNA. Remember, these mitochondrial DNA are extra chromosomal DNA, and they have their own set of replication and help accessory proteins. And on mitochondria also you will have porins, because you need facilitation of movement of materials inside and outside of mitochondria as well. And these granules are these finer structures which where you actually have the biochemical reaction taking place. So what are the functions of mitochondria? So they produce energy through oxidative photophosphorylation. So on the membrane that you just saw, where you saw ATP synthase, you have several other components that facilitate many other biochemical reactions. So for example, in this membrane, you have FADH and this is the ATP synthase. Other than that, you have these complexes which carry the oxidative phosphorylation to generate the ATP. Other than that, you have they regulate the metabolic activity of the cell. They promote growth of new cells and cell multiplication. They have a defined role in apoptosis. So this is the way, so apoptosis is the programmed cell death, and that is also facilitated by mitochondria, where you will have different pathways and different enzymes taking place in which will lead to the disruption of the cells. As in, if the cell has to undergo death because of some damage or because of some problem, then it is the mitochondria that main reactions will take place, where the enzymes will come out from the mitochondria and cleave off the organelles and the nuclear and the cell membrane, so that the cell is finally dissolved. And also it helps in maintaining concentration of calcium and ions within the compartment of cells. And other than that, it will have role in cell signaling, cell senescence, and cellular differentiation. So next we move on to chloroplast, which is a main component of plant cells. The chloroplast is where the photosynthesis will take place. So the chloroplast, their size differs and they are generally in the range of 4 to 6 micrometer in diameter and 1 to 3 micrometer in thickness. And this we already had seen previously, that they are the sites of food producing for plant cells. These are found in the mesophyll cells. They are located in the leaves. So in the leaves, you will see the mesophyll layers, and in the mesophyll cells, you will find this chloroplast. Similar to mitochondria, they have their own DNA and their own proteins. So they carry out their own replication. And these chloroplasts are basically one of the type of the different plastids in the cells. So if the plastids have colors, they will be called chromoplasts. They will give the color to the plant. If they have fat cells, they will be called leucoplasts for the storage of plant fats. But if they have chlorophyll, they will be green in color and they will be called chloroplast, and they will be the site of photosynthesis. So if you see the structure of the chloroplast, you will see again that this is also a double membrane structure like mitochondria. So you will have an outer membrane, then an inner membrane, then you will have the stroma. In this, you will have these stacked components called granum, which will have, each will have a thylakoid. Thylakoid is where the photosynthetic reactions will take place. So if you look at the photosynthesis in a bit more detail, so remember I mentioned about the stroma, I mentioned about the thylakoid. So on the thylakoid, you have these photosystems where the light will be captured, and you have these several components moving across like NADPH and the other hydrogen molecules, which will generate enough of the electromotive force to synthesize ATP, because the ultimate goal is to obtain energy. So you will have this ATP as the final synthesis. So just to go over again, chlorophyll will trap the solar energy and they will then produce this NADPH and oxygen. Oxygen is when there is a, so because of light captured here, the water molecule will be split and oxygen will be generated. The hydrogen molecule and the electrons will be taken in, and then it will be transferred across the several components, and then there will be NADPH synthesis. And because of the movement of hydrogen across these thyroid and stroma, there will be electromotive force which will finally generate the ATP. And the carbon dioxide will be obtained from the Calvin cycle that will be going on along the other side of the mitochondria. Okay, so after chloroplast and mitochondria and nucleus, the next important structures or in the organelles in these cells are the ones which are involved in vesicular trafficking. So vesicular trafficking means something which is produced in the cell has to be moved outside to the cell via secretory method or just transported into the blood veins or the arteries or the capillaries. So you have transportation into, this is from inside to outside the cell, but then you have some components which are on the cell membrane and you need to bring them inside your cell towards the nucleus. So for that also you have organelles which can bring these components from outside to inside the cell. So the main organelles that take part in this kind of trafficking are the endoplasmic reticulum, Golgi apparatus, and lysosome. And there are different methods of endocytosis and exocytosis. So exo means taking out, endo means getting inside. So you can have phagocytosis, which will be like a vacuole will be formed around the component and that will be taken inside in a vacuolar form. Then you have something called pinocytosis. So you will have this pinching of plasma membrane across those molecules, and then there will be a vesicle which will be formed. This vesicle will then be taken forward, and then you have receptor. So you will have some receptor which binds to the ligand, and then these are coated in a vesicle and taken inside into the cell. So this can be carried to the nucleus. So this is for the endocytosis. For the exocytosis, you see there is Golgi bodies here. So the Golgis are responsible for sending the proteins outside the cell. So you will have transport vehicle if it just needs to be transported outside, or if you want to secrete them in some specialized ducts, then you will have a secretory vesicle which will release it into the desired secretion or the tubule. So now the components in details. So we have endoplasmic reticulum. So if you just look at the structure of endoplasmic reticulum, you can see that it will be bound to the nuclear membrane. So the nuclear envelope will be continuous with the endoplasmic reticulum. And they will be again of two types. There will be smooth endoplasmic reticulum and rough endoplasmic reticulum. So the rough means they will have ribosomes attached to them, for example here, and the smooth ones will not have these ribosome constituents. So based on the presence of these ribosomes, they or presence or absence of ribosome, these endoplasmic reticulum will have different functions. So for example, the smooth endoplasmic reticulum, because it will not have ribosome for protein synthesis, they will be involved in the synthesis of lipids like phospholipids, cholesterol, steroid hormones, carbohydrates. But the rough endoplasmic reticulum, which will have ribosomes, will be responsible for protein synthesis and transporting of protein. So remember the dogma. So there is translation, replication, transcription, translation. So the replication is taking place here inside the nucleus, and then you have transcription. So the new DNA is getting transcribed to an RNA here, but the RNA is then taken outside, and here in this site is their translation takes place. So the RNA is translated to a protein here because of the presence of ribosomes. Now, the proteins are synthesized at the endoplasmic reticulum. They now need to be sent to their desired location. So let's say it's a protein that needs to go to the heart region, or it's a protein that needs to go to some other site. It may be just at the epidermis. So it has to be transported from the interior of the cell to the plasma membrane. So that will be facilitated by the Golgi bodies. So these Golgi bodies also have this stack-like structure, and they have this cis phase and the trans phase. So the cis is the one where the proteins that are supposed to be transported will come from the endoplasmic reticulum, and they will be then passed over via the cisternae to the trans phase, where they will be finally pinched off in a vesicle form to transport outside the cell. And similarly, if they are coming from outside the cell into the cell, so they will be fusing here, and then they will be going inside via the cis phase. So the cis phase will then receive materials from endoplasmic reticulum, as we have already mentioned this, and the trans Golgi will be the phase of Golgi towards the plasma membrane, and if that will be responsible for exportation. So here the function of Golgi bodies can be summarized saying that they are responsible for protein sorting. Means where is the protein supposed to go, that will be facilitated by the Golgi bodies, and modification. So while moving across the cisternae, they will have different. So a protein in its native form might not be active until you have some functional group attached to it. So the site of glycosylation, so glycosyl group will be added to it. All these different modification will be done here. And then there is proteolysis. So let's say some of the proteins have not been made properly. It has not folded properly. These proteins are basically of no use. So these kind of proteolysis where these properly folded structure will be dismantled here in the Golgi bodies. And how does that happen? Which are the cells which take place here? So you have enzymes which can lead to proteolysis there, but you have specialized organelle also, where this special structure called lysozyme will be responsible for the death of the cells or degradation of proteins. So for example, the function of lysozymes are the degradation of ingested food material or any pathogenic bacteria or the old protein. So all this digestion or degradation takes place in this cellular organelle, which is called the lysosome, which is also called suicidal bag because it can also lead to autophagy. Now, autophagy is a stage where a cell is starvation in starvation mode, and it cannot survive any further. So it is the lysozyme which will release this hydrolytic enzyme, which will disrupt all the cellular structures. Now, moving to the outside component, we have moved from nucleus to all these vesicular trafficking organelles. Now onto the surface, which is a cell membrane or the plasma membrane. So it is the outermost envelope-like membrane or structure which surrounds the cell. It is also double layered. So you can see there's a phospholipid bilayer here. So this is a phosphate head and there's a lipid structure here. And so if you see here, they will be composed of carbohydrate, proteins, and conjugated molecules. And these are flexible. So there were many models which try to look into the structure of plasma membrane. But the current model that is held is the fluid mosaic model, which says that there are phospholipid, proteins, carbohydrates, and cholesterols which are present, and these are very dynamic in nature because they're interacting with the outside membrane and in the intracellular surface, which makes this plasma membrane very fluid in nature. This was given by Singer and Nicholson. And what are the functionalities of this plasma membrane? They function as physical barrier between the external environment and the internal cell organs. So here, if you see, this is the extracellular matrix and this is the cytoplasm. So this will facilitate movement of the cell or any protein or any component across the plasma membrane. And these are selectively permeable in nature, meaning that only certain molecule will be able to pass from the extracellular matrix into the cell. And as as I have shown in the previous slide, that this is responsible for endocytosis and exocytosis. Here, there will be some proteins which are transmembrane in nature. So they will have a component in the inner part also and the outer part also. And how do that help? They will help in cell signaling. For example, there is some signal to the cell that it has to undergo transcription. So that receptor will be responsible for binding to the ligand and then relaying that information inside the cell. And other than that, it will also anchor the cytoskeleton. So here will be the filament that will be bound to these membrane so that they can give the structural stability to the cells. Now, all this was about plasma membrane and overall cell structure. One of the most important feature about cells are cell cycles. So we'll study about that there now. So what is cell cycle? So if you see a cell, it undergoes several stages during its lifetime. And when it is dividing itself, it undergoes a cyclic procedure so that it can give rise to copies of itself. And that whole process is called cell cycle. This was discovered by Prevost and Dumas when they were studying cleavage of zygote. So this is zygote cleavage. There are several stages. And you can see that from one cell, they have gone to 4 cells and 8 cells. And then there is a whole mass of cell that give rise to the final frog zygote. So they were studying these when they came across the cyclic component of the division. So these are series of stages a cell passes through to divide and produce new cells. And the phases can be divided into mainly two types. Interphase, so the whole phase from this to this is interphase, where the cell is actually preparing itself for division, and then so the preparing itself for division. And then you have the M phase where it is actually dividing and going across all the procedure which leads to its formation of two daughter cells. So the interphase has also 3 cyclical step or stages. So there is G1, which is called the gap 1. Then you have synthesis. Synthesis is important because here is where the DNA replication will take place. And then you have gap 2, where again you have growth and preparation of cells. While in the mitotic phase, it is called M phase because it is the mitotic phase where the mitosis or the cell division will actually take place. And that is also step by step, where it starts with prophase, goes to metaphase, then goes to anaphase, and then there is telophase. So we'll go over these stages one by one because it is important to understand each of them. So first is G1. So start by imagining a cell which has all its nuclear membrane and nuclear components and all the organelles with their own membranes, and then the cell with its own plasma membrane. Now for the cell to divide and become 2, these structures have to be broken apart so that they can literally be divided or distribute themselves in both the daughter cells. So for the first thing or the first phase, the cell first grows physically, copies the organelles, and this makes the molecular building blocks that will lead. So basically, all these organelles, which remember, for example, mitochondria has its own DNA, chloroplast has its own DNA. So they will start replicating themselves, and the organelles also start enlarging and making copies of themselves. Once the cell is now big enough with copies of organelles in themselves, now you will have S phase, which is called the synthetic phase. Now what happens there? You have this DNA, and the DNA will now replicate to form 2N. So here is the step where it will undergo DNA replication. So the DNA content is double. Now what happens? A normal cell is diploid. So in your body right now, any cell that you take from your skin will have 2N as the component, or 2N as the quantity of DNA. Now it will undergo replication. It will become 4N. And that 4N will now be divided equally into the daughter cell, having 2N, 2N each. So that becomes as a normal cell. But right now when it is in S phase, it will have 4N as the quantity of DNA. And with that, the centriole also divides into two centriole pairs in the cells. So if they have centrioles here, the centrioles will also become two, because that we will see when it is responsible for aligning of the DNA. But for now, in the S phase, you have division of centrioles and you have the division or the replication of the DNA. Once DNA has replicated, then you have G2 phase. Now here, the cells grow more, make more proteins and organelles, and begin to reorganize its content for preparation of mitosis. So this phase will end when the mitosis will begin. So here also the cell, because now it has double the content of each, it will start organizing themselves at one end to the other end, so that they can distribute themselves among the two daughter cells. But just before going to the M phase, after G1 phase, if a cell decides not to undergo the synthetic phase, it will enter the G0 phase, or it is also where it will be arrested. So what happens here is, this is a quiescent senescent phase where the cells will remain as the enlarged body for a long time until you have a cue to undergo synthesis. The G0 can be reversible, G0 can be irreversible. So for some cases like neurons, you can have these cells indefinitely in the G0 phase. But for some cells which enter, for example, glial cells, they enter G0 and they remain in G0 for throughout their lifetime. So these are irreversible. They will never enter G1 again or S phase again. But some cells which remain in G0 wait for a cue to enter the S phase. But if there are cells which require faster growing or faster regeneration, then they will skip this G0, and they will, whenever they go into G1 phase, they will complete the entire cycle till the M phase. Now M phases. Now you have to understand this, because this is where your DNA is distributed equally into the two daughter cells. So we'll start with each phase. Keep connecting each phase to the previous phases, so that you have a picture in your mind how the process is actually taking place. So the first step is prophase. Now before this step, there was G2 phase, where you had these double the quantity of DNA and double the amount of all the organelles. Now here you start seeing at the nuclear portion. Now the prophase, the chromosome. Now chromosomes are the DNAs. They are packed together. So these start becoming condensed. So they are packing with each other. They're sticking to each other, coming closer to each other, and they're condensing. And with that, you have the mitotic spindle. These are the mitotic spindle which are outside the nucleus now. And the nucleolus disappears. Why is this happening? Because you want this DNA to attach here, so that when this cell is pinching off from here, you have one set of DNA here and one set of DNA here. But for the DNA to actually go to other cell, it has to first come out of this nuclear membrane. So the nucleolus and the nuclear membrane has to first disintegrate, and that's what's happening in the prophase. And then when it is advancing to metaphase, there is a stage just right towards the end of prophase, which is called the pro-metaphase. So here the chromosomes were already condensing, so they become even more condensed, and they become very compact in structure. And as I mentioned, the nuclear envelope has now broken down. So you can see these DNA or the chromosomes lingering around in the cytoplasm. And the mitotic spindle, which was forming here, they've started moving up towards two different poles. So they have already moved towards these poles, and the capture chromosome at kinetochore. So now just see this. These are the nuclear membrane, and then you have these centrosomes here, which form these microtubules. Now these mitotic spindles or the microtubules will go and bind to the DNA, so that you hold this DNA in one particular orientation. So on the chromosomes, you have something called kinetochore. So this kinetochore will bind to the mitotic spindle. Now we move to the metaphase stage. In the metaphase stage, because you have these spindles right opposite to the spindles are called centrosomes. So you have them opposite to each other, and both of them are attached to one of the DNA chromosome pair. So they align in this equatorial plane, or they align in the center of the cell now. And the two kinetochore, as I've mentioned, they are attached to the microtubules, and they are at the opposite side of the spindle cell. So in metaphase, what happens is, the most important thing is they align at this equatorial plate. Now we'll move on to anaphase. Now, anaphase is when these two duplicated DNA pairs will separate from each other and move towards the opposite poles. So these sister chromatids, they are separated from each other and pulled. So remember the kinetochore? So this kinetochore will be bound, but the junction where the sister chromatids were held to each other will break down, and the sister chromatids will now move towards the opposite sides. So that what happens is at each end it now has its own chromosome, and chromosome was what was required in the cell, right? It was 4n here, and it has got equally distributed in the two halves. So in the telophase, what will happen, you need to start forming these as different cells. So in telophase, the mitotic spindle which was holding these whole DNA and all of them together will break down. So these mitotic spindle are the first to go off. Then what happens is you have this nuclear membrane coming up, which will start forming around these DNA and the chromosomes, and the chromosome becomes to decondense, because remember the chromosome started with condensing, becoming compact. Now it has to go again to the original shape, so it will start to decondense. And then the final step is cytokinesis, basically the formation of two different cells. So you can see that the entire enlarged cells is now pinching off to become two different cells. So this is carried on by two different methods in the two different cells. So in animal cells, it will be by contractile method, while actin is the molecule that will form the cleavage furrow, and the cleavage furrow will chip this cell off. But if you have plant cells, you will have a cell plate which will actually divide this cell into two daughter cells. So you see that if it started with 2n and we ended up from into a 2n stage, but it between, for it to have 2n 2n, it had to first replicate itself to become 4n, which will finally divide into 2n. And that's why, remember, mitosis is called the uh, 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 equatorial division, or cell division in which there are equal halves. Because in other form of cell division, which is called the meiosis, which is called reductional division. So here, instead of being a cell which was from 2n, 2 dividing from to a cell being 2n, in meiosis, a 2n cell will become n. It will have its ploidy half. So that's why it's a reductional division, while mitosis is a equal division. So where are all these cell cycle taking place, and what is the importance? So cell cycle is important for the developmental and growth, as we pointed out at the very first, where which was the basis of the discovery of this cells division. And then you have cell replacement. As I mentioned, there are some cells which require very fast replacement. For example, RBC, and they require replenishment every 13 days, or there are some cells which require replenishment every 4 to 5 days. So for those, you have these cell divisions, you have regeneration. So for example, hydra, if you must be knowing that they are able to regenerate. So if you have any portion which is clipped off from a cell, from an organism, so this cell division is what is responsible for regenerating the lost part, and that is also responsible for their asexual mode of reproduction. Now also, it is not just that it will start cell cycle and go into all these procedures all at the same time. There are checkpoints at every stages, every transition. And what does they do in this checkpoint? So let's say the cell is in G1, it has to move to S phase. There will be a checkpoint right here, just in between G1 and S, which will say if the G1 organelles are in proper sizes, if the DNA is proper there, if it's in 2n shape, if there is any other cellular damage. If there is no damage, only then this cell will proceed to the next stage, otherwise they will not. And how does that happen? There are some proteins, or there's some proteins called cyclin. Your name says they are cyclic in nature. So cyclin E, cyclin A, so there's cyclin A, B, C, D, E basically. So when the cell will have more of cyclin E, it will be G1, which it will be G1 stage. But then at the end, when it is supposed, because it's cyclic in nature, after a certain point of time, cyclin E will go down. And then you have some kinases, which are called cyclin dependent kinase, which are called CDKs. These CDKs too will bind to cyclin E, and they will give a signal for the cell to now move to S phase, because there is enhancement of cyclin A. So once cyclin E goes down, cyclin A goes up. So there is a cyclical flow which is going on. Then here, when you have cycle A changing its CDK partner, it will move to G2 phase. And then you have cyclin B which will come up, and that will take the cell forward to M phase. And then you have cyclin D which will make it come back to, cyclin D will be the one that will start the initiation of the cell cycle. And other than this, these are cyclins, these are CDKs. We also have something called P53. P53 is also a very important checkpoint protein. So here you have P21, P16, P53, which will take care of how the cell structure is organized and how are the DNA damages. If all is good, only then they will allow the cell to proceed to next phase, because it is all to ensure that the new cell or the cell which is undergoing division retains its proper configuration even after division, with all the organelle working in a proper condition. And that's exactly when you have these checkpoints responding as it cannot proceed further, it will either wait at that step and try to correct for its problem, or it will directly enter some reparative method, and if reparation also doesn't work, then it will undergo autophagy and cell death. So that's how a cell's life cycle is maintained. And these were all the topics that were taught in this week. So I've covered mainly all the topics. And if you have still any doubt, you can let me know. You can post it in the discussion forum. We can always answer there. So now, based on these topics, there were some assignments in the previous years. So I'll go over these questions and read the questions, wait for maybe 10 seconds so that you can take a guess, and then reveal the answer and discuss more about them. So the first is the genomic organization in prokaryotes is called what? Is it called nucleosome, nucleoid, chromatin, and chromosome? Then we have which of the following organelle is double membrane? There are many, but you have to be very particular about which one we are talking about here. Endoplasmic reticulum, chloroplast, Golgi complex, and peroxisomes. The bond cleaved by lysozyme in bacterial cell wall between N-acetylglucosamine and L-acetylmuramic acid. Which of these? And the K and the O antigen, which acts as surface associated virulence factors, are present in which of these layers? Lipopolysaccharide layer, lipotecoic acid layer, interperiplasmic space, and outer periplasmic space. So these are the answers, and the first one is very easy because chromatin, chromosome, and nucleosome are very well-defined structure, and well-defined structures are found in eukaryotes and not in prokaryotes. Nucleoid is a very primitive structure, so that's why it is the correct answer here. We have which are the following organelle has double membrane? So we have Golgi, we have endoplasmic reticulum, and peroxisomes. And if you just go over the structure of all the especially Golgi and endoplasmic reticulum that we have discussed in this lecture, you will remember they have continuous membranes but not double membrane. Double membrane is present in chloroplast, in mitochondria, so in nuclear. So you remember from that chloroplast is the correct answer here. Then we have this, which is the bond which is cleaved by lysozyme. If you don't remember the bond which is cleaved by lysozyme, just trying to remember this gram staining protocol that we have discussed, and where we saw that peptidoglycan layer is formed by cross-linking between NAG and NAM by beta-1,4 glycosidic bond. So that's the correct answer here. And then you have the K and O antigen, which acts as surface associated virulence factor, are present in which layer? So for a factor to be responsible for virulence, it has to be present on the surface so that it is able to interact with the host. Now, you need to just remember which of these layer is the most towards the surface. Outer periplasmic, inner periplasmic. Periplasmic says that it is somewhere between. It is a space, so it has to be between two membranes. So this is inner membrane or inner space. Then you have these two contenders, which are called lipoic acid layer and lipopolysaccharide layer. These both layers are definitely present on surface, but which of these will be responsible for containing the antigens? So if you remember lipoic acid layer, these are mainly responsible for adhesion. They are components which help the organisms bind to a particular cell surface. So it is the lipopolysaccharide layer which will have these different carbohydrate and sugar conjugated molecules, which will be the antigens which are recognized by the host, or which are responsible for binding to host. So it will be the lipopolysaccharide layer. The next set of questions are these. So, which of the following species has a linear plasmid? Paracoccus denitrification, Bradyrhizobium ilkani, Brucella canis, and Streptomyces lividans. Then, during plasmid isolation, the reagent used for deproteinization is potassium acetate, phenylchloroform isoamyl alcohol, sodium acetate, or zero point 0.2 molar NaOH and 1% SDS. And the biomolecule synthesized inside the nucleus is histone, DNA polymerase, tRNA, and RNA polymerase. And the major protein that cause condensation of eukaryotic chromatin are RNA polymerase, DNA polymerase, histones, and ribosomes. So in the lower set of questions, remember the functionality of the proteins and what is the nature of the component that is written, and then you will be able to understand where can these biomolecules be synthesized. So these are the answers. So we'll go over them one by one. The first one is, which of these species are the linear plasmid? So as I was talking, when I was talking about plasmid, I had mentioned that they could be linear or circular in nature. So Streptomyces lividans has a linear plasmid. And then during plasmid isolation, the reagent used for deproteinization is which of the following? And the answer is phenylchloroform isoamyl alcohol, because in the other reagents, they have their own specific purpose. For example, potassium acetate and sodium acetate are used for precipitation of DNA, to get the DNA. So to bind to the DNA and get it out of the solution. And 0.2 normal NaOH and 1% SDS are for the lysis of cell. So deproteinization basically means removal of the proteins and getting all the debris out of the DNA and just having the DNA detention. So this will be combined effect of phenylchloroform isoamyl alcohol, where you can separate the DNA out of all the other macromolecules in the lysate. And then you have biomolecules which are synthesized inside the nucleus. So inside the nucleus, you have transcription and you have replication. So histones, polymerase, and this RNA polymerase, they are proteins. So they have to be outside the cell at the endoplasmic reticulum to finally give their proper fold. But tRNA is a RNA which is mainly part of transferring of the amino acid during the translation process. So this is an RNA compound. That's why it is synthesized inside the nucleus. And then you have proteins that can cause condensation of eukaryotic chromatin. You have RNA polymerase, whose function is to translate from RNA to protein. You have DNA polymerase, which synthesizes copy of DNA. You have ribosome involved in the synthesis of protein. But you have histones, which are compositional unit of DNA, and these are responsible for condensation of the chromatins. So that's why histones. Okay, the next set of questions. The cellular organelle that cannot be obtained intact during fractionation is endoplasmic reticulum, lysosome, peroxisome, or nucleus. The phase of Golgi complex that sorts lipids and proteins is called. The orientation of the protein in plasma membrane is predetermined by their orientation on the endoplasmic reticulum membrane, nuclear membrane, mitochondrial membrane, or vacuolar membrane. And the membrane carbohydrates are exclusively confined to which side of the plasma membrane. So it is cytoplasmic phase, exoplasmic phase, core of the biolayer, or both sides of the cytoplasmic or exoplasmic phase. Try to imagine the organelle and the sides, and you can come up or you can think where the possible proteins and lipids will be placed. So these are the answers. So the first is the cellular organelle that cannot be obtained intact during fractionation is which are the following. So if you look at the size of these organelles, you have lysosome, peroxisome, and nucleus. These are one singular units, and these can be obtained. So the process that we are mentioning is fractionation, which means the cell will be under very high pressure, or it will be subjected to very high pressure so that it can fractionate the cell. Now, lysosome, peroxisome, and nucleus are smaller structures when compared to endoplasmic reticulum, which is this big contiguous organelle in continuation with the nuclear membrane. So there are very high chances that during fractionation you get fractions of the endoplasmic reticulum itself, because lysosome are structured one organelle that can come out on itself. Peroxisome is similar, and nucleus, you can have nucleus isolated from the cytoplasm, but you have endoplasmic reticulum, which is this big whole structure that can get fractionated when you are putting the cell under very high vibration or subjecting them to very tough conditions for extractions. Okay, the next is the phase of Golgi complex that sorts lipids and protein. Now, sort lipids and protein, cis phase. The cis phase, you remember, that is towards the nuclear membrane. So that's the proteins and lipids have to go in and they are sorted there, because if it is protein and it has to move out, it will be on the outer surface. But they are sorting it, and the sorting is at the so trans is at the exact near the plasma membrane. It's the cis phase which is at the end. So the protein sorting takes place at the behind, which is the cis phase. And then you have the orientation of the protein in the plasma membrane is predetermined by the orientation where. So which is the membrane, which is the organelle which is responsible for trafficking the proteins or carrying of these proteins in endoplasmic reticulum. That's where the orientation, which part of the protein will be outside, which will be in the intermembrane surface, and which will at the cytoplasmic end, is determined at while it is being trafficked from the nucleus to the plasma membrane. So it is the endoplasmic reticulum where the orientation is determined. And then you have the membrane carbohydrates are exclusively confined to which side of the plasma membrane? Cytoplasmic phase, exoplasmic phase, core of the bilayer, and both sides of the cytoplasmic and exoplasmic phase. So you need to remember that carbohydrates are responsible for attaching to several lipids and several other proteins to carry out signals and other information for the cells. So it has to be outside onto the surface facing the extracellular matrix so that it can bind to these proteins or other sugars. So that's why it's on the exoplasmic phase. So from the phase which is outside the cytoplasm. The next set of questions are the marker enzyme responsible for lysozyme is uric acid, cytochrome oxidase, stearic acid transferase, and cathepsin. The name suggests the functionality, so try to think. Then you have transferrin, a soluble carrier protein that transports iron through the blood by a process called phagocytosis, cytosis, receptor-mediated endocytosis, and exocytosis. Again, think on the term and think of the functionality of the term so that you can understand what does this question, how it can be interpreted. And then you have the lumen pH of late endosome, where the contents are digested and delivered for further distribution. So you need to understand the pH, but remember the stage where it is talking about, which stage we are talking about. And the decision to divide is taken during which phase of the cell cycle, G0, G1, G2, or S. So these are the answers. So we'll go over one of each of them one by one. The first is marker enzyme for lysosome. So if you didn't even know, if you just go over the name as I suggested, it will be giving you an idea of what kind of component it is. The first is a uric acid, and it can't be an enzyme because it is acid, and you have an idea that uric acid is mainly involved in bile composition and urea synthesis and all that. Then you have cytochrome oxidase. You have cytochrome oxidase on the mitochondrial membrane because there it will be important for undergoing the oxidation processes. Then you have called something sialic acid transferase. The name said it is for sialic acid transferases. So then what is left with cathepsin, which is for the lysozyme marker enzyme. Then you have transferrin, which is again a soluble protein which transports some iron throughout the blood by process of what? So transferrin is a receptor, and if it is carrying something via vesicular trafficking, then it has to be via receptor mediated endocytosis because it is getting something inside the cell and not phagocytosis, because phagocytosis and pinocytosis just engulf the compound or the whatever molecule they have to insert, and then they will form vesicle or a vacuole. So there is no role of any other protein there. And exocytosis is not correct because the blood is being carried inside the cell. So and transferrin is something which is transporting inside to the cell. So this is an endocytosis process. That's why it's receptor mediated endocytosis. The next set of question is the lumen pH of late endosome, where the contents are digested and delivered for further distribution. So, like we had mentioned that the endosomes or the lysosomes will have acids which will break down the components. But now we are talking about late endosome where you have already digested the contents and they will be now distributed further. So to like excrete the damaged material. So the acidic pH will go higher because of these release of these components from the digested material. So the pH now increases, not neutral, but to a certain extent. So that's why it's 6 to 6.2. And then we have the decision to divide in is taken in which during which phase of the cell. So we remember G0 is quiescent phase, there is no division there. G1 is the first stage where it will actually undergo all these cell organelle duplication and the cell enlargement. So this is the stage where the cell will decide that it has to undergo a cell cycle and a division. So these are the next set of questions, which are histones. They are synthesized in which phase? M phase, G0, G2S. Remember, where are histones important and where will they be synthesized? The cells which do not divide once they are differentiated and enter G0 are of which of these cells? Lens, lymphocyte, epithelial, and smooth muscle cells. During which phase of the cell cycle the chromosomes becomes highly condensed to prevent DNA damage and facilitate segregation to opposite poles? Prophase, metaphase, anaphase, telophase. And the pH in mammalian cell culture is monitored by waste. So this is a very experimental question, but this is important if you do experiments or you need to know basics. So Texas red, phenol red, crystal violet, trypan blue. So these are the answers. So histones are synthesized in which phase? So remember, histones are responsible for the condensation of the DNA. So they will be mostly important for the S phase. So that's why they are synthesized there, synthetic phase. Hmm. The cells which do not divide once they are differentiated enter into G0 phase. So we'll go over each of these cells and see if they are supposed to divide or not. We have smooth muscle cells which divides. These cells do divide. Epithelial cells divide very faster, at a very faster rate because they have to replenish the intestinal layers continuously. Lymphocytes, we know they are responsible for immune responses, so they are continuously under the process of cell division. So it is the lens cell which enter G0 phase. Then we have during which phase of the cell cycle the chromosome become highly condensed to prevent DNA damage and facilitate segregation to opposite phase. So metaphase, they are arranged, but it is in anaphase that they finally move to the opposite pole at a higher condensed rate because in metaphase it is still condensing. And then last is the pH in mammalian cell culture is monitored by which of this? So the answer to this is phenol red. Crystal violet is another dye that we have seen is used in the gram staining procedures. And trypan blue and hexazide are also dyes, but they have their own. These are used in different other cultures or different other experiments. So this covers for the questions that have been done in the previous years. But if you have any of questions, you can definitely type in the comment section or discussion forums. And that covers the topic for today. I'll just go over these slides once again from the start so that we can go over. I'll just give you a summary of each of the slides so that you can recapture what we have studied so far. Yeah. So the topics that we covered in this presentation are cell biology to understand cells and to look at the cell division or cell cycle. So we started with what is cell and how our cell is structured and what are the components of cell. We then went over how was a cell described, right from just looking at it as a simple room-like structure to actually looking into the structural components of the cells, which has nucleus. And then based on all these discoveries and what was finally looked into via different experiments, we saw the characteristics of cells, like what are the components that are present in the cells, and we went over each of them separately. So this is just a brief overview of how the cell looks like. We have gone over mitochondria, cytoplasm, plasma membrane, lysosome, endoplasmic reticulum, and ribosomes and Golgi apparatus. But what we haven't looked into are the finer details like microtubules. Centrosomes we have spoken about when we were talking about in the cell cycle. Microfilaments are also responsible for movement across the cell. We have vacuoles, which have these digested proteins or any of the engulfed material. We have talked about secretory vesicle when we were talking about trafficking. We have nucleus, nucleolus, part of the nuclear membranes and nuclear structures. So we have spoken about all of them in somehow or the other details. Then we looked into the differences between prokaryotic versus eukaryotic cells, how they differ in several aspects like size, the presence of nucleus, well-defined membrane-bound organelles, flagella, the cell wall, plasma membrane, the components, the constituents of ribosomes, and how do they divide and what are the mode of sexual reproduction. We then went over prokaryotic cells, mentioning their features and what all they lack, how they have only some basic structure of nucleus, which is called nucleoid. And we defined each of these structure and their functions individually. We then went over a very common example or common experiment, which is called gram staining, and we saw what is the basic differences between these two kind of cell types, which are called gram positive and gram negative, which is where the theory or the idea of the staining protocol lies. And then we saw how the experiment is actually carried out and how do we interpret the results that we see in the slide. Then we went over one of the very important constituent of the bacterial cell, which is called plasmid. Why it is important not only for the bacteria but also for applied purposes like cloning. And why do we do cloning using plasmids? What is the important feature of it? And what are the different forms of plasmid in the bacterial cells? And why do the bacteria cells keep plasmid? And why? What is the importance? We move from prokaryotic to eukaryotic cells. We've defined that the differentiating feature about eukaryotic membrane is the cells is the presence of nucleus and membrane-bound organelles, and then eukaryotic cells themselves can also be divided into two types: plant cells and animal cells. We again went over how are these different from each other, mainly on the basis of presence of the vacuoles and the cell wall. And then we went over each of the components of the eukaryotic cells, like cytosol. Cytosol is different from cytoplasm, though they are correlated to each other, but it is important to know the finer details and the differences between them. Then we went to nucleus. We understood the structure of nucleus, and then we also looked at the nuclear envelope, which is important for the transport of material into and outside the nucleus. Then we went to the powerhouse of the cell or mitochondria. We went over the structure of the cells, structure of the mitochondria, and how that is responsible for several of the important processes that take place in the mitochondrial membrane, which is ATP synthesis or oxidative phosphorylation or the apoptosis. Then we went to chloroplast, which is a type of plastid, and what are their general features and their structures, and where does the photosynthesis actually take place in the chloroplast. And then we went over photosynthesis as well to understand these steps involved and how they are placed in the chloroplast. Then we looked into the organelles which are responsible for trafficking or movement of materials throughout the cells. This was for endocytosis, which were of three types: phagocytosis, pinocytosis, and endocytosis via receptor mediation. And then we also went over exocytosis, which is the movement of materials outside the cell. And we saw each of these organelles which are involved separately. The first, we looked into endoplasmic reticulum, which will be associated with the nuclear membrane, and they will have ribosomes attached to them. If they are with ribosome, they are called rough endoplasmic reticulum. If they are not with ribosome, they are called smooth endoplasmic reticulum. And the basis of their functionality is determined by the presence of ribosomes. So if they have ribosomes, they are involved in protein synthesis. If they do not have ribosome, they are involved in lipid synthesis or phospholipid synthesis. Then for Golgi bodies, we saw that how are the cis phase and the trans phase arranged, and how to move, but how are the orientation determined during the movement, and what are the roles or other than the functions of the Golgi bodies across its structured component. And then the third component was lysosome, where we saw what is the component that actually carries out the degradation of the protein, which is the hydrolytic enzymes or the lysozyme, and what are the functions and what are they mainly used for in the cell. And then we go to the external most structure of the cell or the component of cell, which is the plasma membrane. And we went over the structure where we saw that they are phospholipid bilayer, they're fluidic in nature, and they have several conjugated carbohydrate and glycolipid, which is responsible for the interaction with the extracellular matrix and the cytoplasmic part. And then based on this introduction, they have different functions. For example, they act as a physical barrier, they're responsible for movement of materials, they are semi-permeable nature, so they have very categorically they move materials in and out of the cells. And then they are also responsible for cell signaling and endocytosis and exocytosis. Then after defining cells and their components, we move to cell cycle, where we first saw what are the general class stages of cell cycle, which are called the interphase and the mitotic phase. Interphase will have gap 1, gap 2, and synthetic phase. And a mitosis phase is the phase where the cell actually undergoes division, which is again divided into 4 stages: prophase, metaphase, anaphase, and telophase. And then we went over each of these phases one by one. We had G1 phase, S and G2 phase, which are more like the preparatory phase for the cell to undergo division. So in all these stages, the organelles duplicate, the cell grows in size, the DNA duplication happens, and then the cell enters mitosis phase. But just before entering the mitotic phase, it can also enter into G0 phase and remain quiescent or non-proliferative for a long period of time. And G0 can be reversible or irreversible based on the Q and the type of the cell. And then we went to mitotic phase, and we went over each of the different phases, which can be prophase or pro-metaphase and metaphase. We started with the fact that how the DNA condenses, nuclear membrane disappears, and how they're aligned at the equatorial plate at the metaphase, connected to the mitotic spindles via the kinetochores. And then we move to the next phases where the DNA, the kinetochores are able to move the sister chromatids to the opposite pole because the linkage of the proteins which held these sister chromatids with each other have all broken off. And then at telophase, both the poles will have their own chromosomes and the nuclear membrane starts reappearing. And the cytokinesis being the final stage where we will have the pinching off of the new cells from each other. And then we went to how this cell cycle actually functions and what is the role of this in a particular organism, where we saw that it could be for development and growth, which was how the cell cycle was discovered. And then we have cell replacement, basically cells which have which require replacement in very shorter period of time. They undergo this at a very faster rate as compared to the one which are entering G naught phase. We have regeneration and asexual reproduction, which are mainly driven by such cell division. And then we came to a very important factor, which is called the control of cell cycle, where we went over which of these components are responsible for proceeding or giving the cue to the cell to proceed to the next stage. So we have cell cycle points, which are used for completion of different steps and repair of DNA damage. So here they determine if this cell at a particular stage is suited to move into the next stage. And these points are called checkpoints, and they are many molecules, proteins which are constituents of checkpoints. They can be CDKs, they can be cyclins, and they can be P51 and P53 and P21. So all these together make sure that the cell is properly arranged and structured, and all the components of the cells are in proper shape during and its cell division and it's during its lifetime as well. So these were the topics that we had covered, and then we went over several questions from previous year assignments, and we discussed each of the answers in details. And we saw how we also tried to reason out if there were any new kind of question and how to proceed with these questions. So with this, I'd like to finish this presentation. The presentation ends here, but if you have any question, you can definitely put it in the comment section or in the discussion forum of your course, or you can even email us. We're always happy to help. Thank you.